Good afternoon, everyone. Thank you for coming on time. And thank you for accommodating me to do online learning today. So let's get started. First, we will begin with a little bit of review of what we've learned last Monday. Hold on, let me turn on the live caption for anybody who might need that. There we go. Okay, so we're going to start with a little bit of review from last class, last Monday, actually, when we learned about the process of DNA replication. So please turn to the last page of your study guide and you will see a picture like this. So just to reorient you, this is one side of the replication bubble. Okay, so if you remember, this is the parent DNA, and then you're going to open up a little bubble, right? And then we are going to be focusing on one side of the bubble. That's what this is, the replication fork. And we have several enzymes here along the way. I'm just going to help you label it, and hopefully it will refresh your memory about the process. So first of all, we have an enzyme right here, number 2. Okay, this enzyme is responsible for unwinding the DNA, creating the replication bubble, and continuing to open up the DNA. That enzyme is helicase. Okay, so you would have a helicase. You will have a helicase over here on one side of the fork that's going this way, and you would have another helicase on the other side. And as they move away from each other, they're going to be opening up the double helix. So you can gain access to the nucleotides. Now, as the helicase is unzipping the DNA, it is going to create a bit of tension that builds up within the DNA double strand. And we must relieve those tension to prevent DNA from breaking apart. And that will be the job of this enzyme right here, this particular protein. As helicase unwinds, this one is going to relieve the tension. Does anybody remember the name of this enzyme?
Parental DNA.
Okay, so this is showing the parental DNA, Colleen. That's correct. But what's the enzyme that will relieve the tension? Anybody remember that? That's right. That is the gyrase. Gyrase. Okay, so just to reiterate, as helicase unzips the DNA, you need to relieve the tension. The gyrase does that. The gyrase will reduce the tension, so the DNA is not going to break. Now, the DNA strands, the parent strands, they would have a tendency to reconnect together. They want to come back together because the nitrogen base are attracted to each other, and we certainly want to keep them apart, and that's what these little circles are going to do. Right? We also label them in the note that we took on Monday. These are single strand binding proteins. Okay, they prevent reannealing from happening, prevent the strands from coming back together. You keep them apart. Okay. Now I'm going to label several things at the same time here. Remember that DNA polymerase 3, it has 2 limitations, right? Number 1, it cannot initiate the process of replication. It doesn't know where to start. Therefore, we must put a few base pair there to guide the process of replication, right? And so it's represented by these red things here. And so we will put one right here, put one right here, put one here, put one here. So all these red stuff that I'm drawing here, those are the RNA primer. RNA primer. And again, the function of the RNA primer is to guide the DNA polymerase 3 so that it knows where to start. So once you put the RNA primer there, then you can continue with the process of replication, right? On one of the strands, you only need one primer. But on the other one, the lagging strand, you will need to have multiple primers, right? The more you open up, I'm going to need to put another primer and another primer and so on and so forth. Now we need to have an enzyme to do all this. We need to have an enzyme to create the RNA primer, and that's shown here by this square here. I'm not going to ask you to label this diagram on a test or quiz, right? Like this is just for summarizing the information. So it's kind of, they all look just like ovals and squares. There's no point memorizing it, okay? But it's a good idea to use this picture to, help you organize all the information. So what's this enzyme here? Does anybody remember? The enzyme that will help put the RNA primer in place. What is the name of that enzyme that does that? Okay, that's fine. This would be the RNA primase. Okay, RNA primase. The primase will put the primer. And again, the purpose of the primer is to help position the DNA polymerase 3, right? So that it knows where to start. Remember in class I said, it's like my kids, right? They don't know how to do their homework. So I had to do a few questions for them, with them, and then they will be able to continue on their own. So that's the function of the primer. To put the primer there, you need the primase. Okay, so once the primer is in place, then we will be able to continue to build the strand, the daughter strand, in the 5' to 3' direction. Right, 5' to 3', like that. Okay, and the enzyme that allows you to build the DNA strands is represented by this guy right here, right? And you have another one here and another one here. They all represent the same thing. That would be the DNA polymerase number 3. Okay. So the strand over here that is being replicated continuously with no breaks, that is the leading strand. And you might remember that name, leading strand. And it's being created continuously. Okay. And the direction is 5' to 3'. It's always 5' to 3'. And it's moving towards the fork. Right? It's going to go into the fork. As helicase unzip more, you will just keep on going until the very end. On the other hand here, this strand that is being synthesized discontinuously, that would be the lagging strand. Lagging strand. Okay. Discontinuous. Discontinuously. Still, it's 5' to 3'. It's not going backwards or anything. And it's away from the fork. The fork. Okay. All these fragments, you might remember the name, right? The Japanese scientist, Okazaki. Fragments. Okay. You have all these already, right? We did it on Monday, but we're just refreshing your memory because we didn't have a class on Wednesday, or we had the lab, and so it's good to just bring you up to speed. Now, very quickly, the next part is hard to show on the diagram, but in class I was saying, as the DNA polymerase 3, the green oval here, approaches the primer, then it's going to fall off, right? So after polymerase 3 falls off, which one comes on? Do you guys remember? After number 3 falls off, what enzyme is going to take its place? If you're thinking about DNA polymerase 1, that would be correct. Okay, so after number 3 falls off, number 1 will take its place. And the job of DNA polymerase 1 is to remove all these primers and replace it with actual DNA. Okay. And afterwards, we have to stitch all the fragments together. And that will be the job of the ligase over here. I'm running out of color, but there is the ligase. Okay, DNA ligase. Okay, so that's sort of where we left things on Monday, and we're going to continue with the PowerPoint. If you have any questions, right, just unmute and ask away, or if you're a little bit shy, just type the question in the chat box. Okay, couple of extra things here that's worth mentioning in this slide. Like I said, DNA polymerase 1 will remove the RNA primers and replace them with the appropriate nucleotides. The only one that's really worth replacing is the uracil, right? You take away the uracil and replace it with thymine. Everything else is analogous in RNA and DNA, right? It will remove the adenine, but then it will just put the adenine back on. It's only the uracil that really needs to be replaced. The ligase will join the gaps in the Okazaki fragments. Now, this next point I didn't really mention in class last time, but both the polymerase 1 and 3, they have proofreading capabilities. So as they are creating new strands, they will also be checking errors along the way. It's like the spell check on your word processor. Okay, if you make a spelling mistake, it underlines it with the red squiggly, right? And then you will know to press backspace and then type in the correct spelling or something, right? So that's exactly the same thing with DNA polymerase 1 and 3. If it detects an error, right? Let's say it was supposed to be a G and then it accidentally pairs it with a T instead of a C. It will go back, delete the T, and replace it with the correct one. Okay. Sometimes despite its best proofreading effort, it still end up making a mistake. And if that mistake is not corrected, it will result in a permanent DNA change. And we call that a mutation. So when it comes to mutation, let's go back here for a second. When it comes to mutation, there are two groups of cells in our body. And you should write this down because we can come back to using these terms quite a lot for the remaining of the semester. We have cells that we call somatic cells. Somatic cells. So these are basically all the cells in the body. All the cells in our body except for the sex cells. Okay, so sperm and egg excluded, everything else is somatic cell. The germ cells is the other group of cells that we have. Now, this is a little bit confusing for a lot of people. They hear the word germ and they think of like, dirty stuff like bacteria and, viruses. That's not what it means, okay? Germ cells in the context of what we're talking about is referring to cells that will become sperm and egg. Okay, so it has nothing to do with bacteria. These are just cells that will become the sex cells, the sperm and the eggs. Now, if there is a mutation in our somatic cells, then it will only affect you alone, right? The individual who has the mutation in the somatic cells is the only one who will be affected by it. However, if a mutation occurs in the germ cells, then future generations would be affected by that mutation because the sperm and eggs will be the cells that are used to create the next generation, right? You might have heard of gene therapy, and we'll talk about this in lecture 9. When we try to do gene therapy, there is an ethical debate about whether we should be permitted to do gene therapy on germ cells. If you do gene therapy on somatic cells, only that particular individual would be affected by the therapy. Okay, but if you do it in germ cells, then all future generations would potentially be affected, and that raises some serious ethical concerns. But for now, we'll leave it at that. Okay, does everybody understand the difference between somatic cells and germ cells? There is a little like an icon in the chat box that you can click to give like thumbs up and stuff like that. All right, so you can use that to give me an idea of whether or not you are with me or not. Okay, because it's hard to tell. In class, I can see your faces and I can see whether people are with me or not, but it's harder to tell here. Okay, so if you can just give me some visual response using the little thumbs up icon and whatnot, that would be good. Do you guys know what I'm talking about? Do you guys use the thumbs up thing in math class or in other online setting? No. I'm not seeing any thumbs up. So oh, I see one. Very good. Right, so if you know what the button is, please use that to just give me some visual feedback so I know you're with me. Thank you. Okay, so moving on. This is the ligase, the one that will connect the gaps, join the gaps together, and to complete the process. So everything we talked about so far, that is DNA replication in prokaryotic cells, right? In simple cells like E. coli and like other bacteria. But the process is very similar to what happens in our body, very similar to what happens in a eukaryotic cells. But there are a few subtle differences, okay? And I'm going to highlight 3 key differences here. Number 1. Eukaryotic cells have linear chromosomes as opposed to circular chromosomes. If you remember, long time ago, we drew a picture of a bacterium, and I showed you that there are these circular DNAs in the center, and then there's some plasmid. So all of the genetic material in a bacteria is circular, right? And they also don't have as many nucleotides as we humans do. And so for them, it's okay to have a single origin of replication. But for us, in order for the process to be efficient, we would have multiple origin of replication. Okay, and it's not uncommon to have up to hundreds of thousands of origin of replications in a human cell. So this is what it looks like if you have multiple replication bubbles. You will create them, and then they will expand laterally, they'll expand horizontally, and eventually fuse with each another. That just kind of speed up the whole process of DNA replication. So that's difference number 1. Difference number 2, most of the enzymes in prokaryotes have a eukaryote counterparts. Okay, so you saw that we had the RNA primase in the prokaryotes. We also have that as humans. However, some of the enzymes are represented by multiple versions of the same thing. For example, in humans, the DNA polymerase comes in 5 different versions. It's not just 3 and 1 like the E. coli. Okay, there are multiple other versions as well. And most of them have overlapping functions, and it's just to make the process more efficient and faster. Okay, so those are the 2 main differences that we talked about so far. And then the third one is that some DNA are lost during each DNA replication in eukaryotic cells. This is true, and a lot of people might not know that. Every time our cell copies the DNA, we will lose a little bit of it. Okay, so let's say you start with DNA that's this long. After one division, it gets shorter, and then shorter and shorter and shorter and shorter. So after each division, you will keep on losing a little bit of DNA every time. Now, you might wonder, why? Why does that happen? And how come it doesn't happen in the prokaryotes, but it happens in us, in humans? Well, I'm going to try to explain that to you. You can draw along if you want, or you can just watch. This is being recorded anyways, so you can come back and listen to it again and try to draw it out again at a later time. Okay, so let me just go through some of the basic things again. I'm going to do this quite quickly because we have reviewed it already. Let's say this is 5' and 3'. And on the other side, it will be 3' to 5', right? Because DNA is anti-parallel to each other. Okay? So another thing we talked about was when you open the replication bubble, we're going to put a primer here, and then we're going to put a primer like here. And with the DNA polymerase 3, it's going to come, and then it will replicate in this direction, right? Okay, because if this is 3', This would be 5 prime to 3 prime. That's nothing new. That's exactly what we talked about earlier. Okay, and the same thing here is going to go this way. But when you open up more of the fork, right, because the polymerase 3 cannot synthesize backwards, right? So you must put an other primer, right? And you must put an other primer and another primer. And so this is going to go forward. This is going to go forward. This is going to go forward. Right? That's everything we know so far. Nothing is new. And then what happens is when the polymerase 3 gets to this point, gets to the primer, it's going to fall off. And then polymerase 1 will come. And polymerase 1 will basically replace that with DNA, right? This thing, same thing, it will get replaced with DNA, right? This part will get replaced with DNA. However, this part will never get replaced. There are no polymerase coming from upstream. The primer will not be replaced. Okay, so this chunk of DNA actually never get copied, right? The primer would just fall off, and then that little bit of DNA would be lost in the new one, okay? In the new copied version. Same thing over here. Okay, so this is fine. This goes all the way to the end. Everything is copied. But you know, as you open up more of the fork on the other side, you're gonna need another primer and another primer and another primer. Right? So polymerase 3 will go this way, and then this will get replaced. This will get replaced. This will get replaced. But same story here, right? This will be lost. Will not be copied. Will be lost. Because there's no polymerase coming from upstream, right? From before it. So the primers will not be replaced, and then it will just be gone. Does that make sense? I need you to understand this part because a lot of what I'm about to explain in the following slides has to do with this particular concept. Just gonna pause for a moment and see if anybody wants to ask anything.
Can I ask you a question?
Please go ahead.
So is it just the RNA primers on the 3 prime? Those are the only ones that won't change.
That's right. Okay, but here's the thing. I kind of understand what you are really asking. Let's say this is the original one, right? Okay. So afterwards, you will copy everything, but missing a little bit here, right?
Yeah.
And then same thing on the other side, you will copy everything, and then you will be missing a little bit here, right? But when this strand joins with this strand, because this part here cannot find like a complementary pair, this part will actually be lost as well. And then same thing over here. Even though you only have one side of it, because you cannot find the other's complementary part, this part will be gone, and so it will become shorter.
So they just match to each other.
That's right. And then overall, the blue ones are shorter than the black ones, right?
Okay.
Okay. Now, another question that you might have in the back of your mind is, how come this is not a problem for bacteria? Well, how come it's not a problem for the prokaryotic cells? Well, prokaryotic cells, they have circular DNA, right? And just for simplicity, I'm just going to draw one circle as opposed to the double helix. So they will put a primer here, and it will go and replicate itself, and it will always be able to loop back to where it started. And so it will be able to replace the primer. There will always be something coming from upstream because it's a loop, right? We have linear DNA. That's why we don't have the loop. That's why we cannot replicate the ends, because nothing is coming from upstream. So that's why circular DNA will not have this problem. Okay, hopefully that makes sense. Feel free to ask more questions if you think of any later on. So how do we solve that problem? We don't want to be losing DNA every time we divide. That could be a problem. For example, if you think about like the whole DNA here, Okay, oops. Think about a whole DNA section here. You might have genes here. So that might maybe like gene 1, you might have gene 2 here, gene 3, so on and so forth, right? You might have a lot of genes. So if you keep on shortening your DNA, eventually you're gonna lose some of your genes, right? Genes are a segment of DNA that encodes for proteins, right? And if we damage those genes, we're not going to be able to make the proteins that they code for, and that could be bad news. So how do we protect them? Well, it turns out at the ends of your linear chromosomes, you have what's called telomeres. Okay, telomeres. And telomeres are repeats of TTA, GGG. So you will have a bunch of TTA, GGG located at the end. Okay, this is obviously not to scale. You know, it's the actual chromosome is much longer, and then you will have these things repeated like hundreds and hundreds of times. Right. So what happens when you do DNA replication? It gets shorter, but you will just be cutting off a little bit of your telomeres at a time. Okay, so next time you divide, you'll cut a little bit more, right? And then you cut a little bit more, right? So you're not actually losing important genetic information. You're just cutting things at the end that don't really code for anything. Okay, you're just cutting off all these things that are repeats of TTA, GGG. So the purpose of having these telomeres on both ends of your chromosome is to delay, to postpone the erosion of genes that are located at the ends of the DNA. Without these telomeres, you would eat into the genes very quickly, right? And you might end up losing important genes. So that's why we want to have the telomeres. In this picture, you can see the part that lights up at the very end. That's where the telomeres are located. Okay, and you will find them on both ends of the chromosomes. They're kind of like the plastic thing that are located at the end of your shoelaces. They are there to protect the shoelace, right? So that it doesn't fray, it doesn't like unwound and break apart. And that's exactly what the telomeres do. So typically speaking, a cell can divide maybe 40 times or so. And afterwards, the telomeres at the end, they would become critically short. Okay, that means by the time the cell have divided 40 times or so, there will be very, very little telomeres left. If you keep on dividing beyond that point, you will eat into the important genes. So we should not allow the cell to divide any longer. When telomeres are critically short, the cells will no longer divide. Okay, so the cell is going to enter a senescence state. Okay, so this is like when a cell is too old, too many divisions have had happen, and they are not gonna be able to divide again. Okay, so when I'm just gonna put an arrow here. Okay, when telomeres become critically short, the cell can no longer divide, and they enter a senescence state. When there are a lot of cells in your body that have entered senescence, that cause you to age. Okay, so it's part of the aging process. That's one of the theory, anyways. Okay, why do we get old, right? Why do we have a reduced ability to heal wounds and so on and so forth as we get older? That's because there would be a larger pool of cells that have entered senescence. Okay. So everybody clear on what telomeres are and how they protect our chromosomes? Any questions? I'm still not done talking about this thing, but I just want to make sure we're all clear up to this point. Okay. Thank you. Do you guys know what Dolly is? Or who, what, I guess what? Do you guys know what Dolly was? Anybody have heard of Dolly before, maybe? So Dolly was the first mammal to be cloned from an adult somatic cells, using the process of nuclear transfer. So I'm just going to try to explain a simplified version of what happened here. In order to clone Dolly, they needed 3 separate sheep. Okay, so we would have a sheep that is going to be the egg donor. Okay, so they extract an egg from the donor, and then they remove the nucleus. Remove nucleus. Okay, so it's just an empty egg with no nucleus. And then they have to have a cell donor from an other sheep. Okay, so they would get a, sorry, this is egg, yeah, and this would be a somatic cell. Somatic cell. And I think they took it from the udder. And what happens is they will extract the nucleus. Extract nucleus. Okay, so they take away everything, keep the nucleus intact. Then they put the nucleus into the empty egg. And then you end up having an egg with 46 chromosomes in it. Not 46, with a full set of chromosomes. I forgot how many chromosomes the sheep has. Okay, but the egg would only have half of it, right? So they remove the nucleus, and now they put the nucleus of an adult cell into the egg. So now it has the full set of chromosomes. Then they implant this egg with the full set of chromosome into a surrogate mother. Surrogate mother. Okay, and then let the egg grow in the uterus. Then what happens is at the end, the surrogate mother gave birth to Dolly, which was a genetically identical sheep as the cell donor. Okay, so this Dolly is a clone of the cell donor, not the clone of the egg, because the egg donor was just providing the egg, where there's no genetic material, right? They removed the nucleus, not the surrogate mother, because the surrogate mother, as the name implies, is just the sheep that provide the womb for the sheep to grow, right? So this is genetically identical to the cell donor. So far, hopefully you're with me. And what happened was the sheep, Dolly, was only able to live for 6 years, half of the expected lifespan. Right? So here is what happened. Here is why the sheep was not able to live, a healthy, long life. Okay, that's because of the telomeres. Okay? So I was telling you, every time we divide, the telomeres get shorter and shorter and shorter. Eventually, they get critically short, and they're not allowed to divide anymore, right? Okay. So the donor, the cell donor sheep, was an adult sheep. So by the time they extract the cell from this donor, the telomeres would have shortened since the birth of that sheep, right? So let's just say, for the sake of discussion, the donor, the cell donor, is like 10 years old. Okay, 10 years old. Okay, and that's like adult for a sheep, right? Okay, so that means the cellular age is 10 years old. That means even though when Dolly was born, it's like, newborn baby, age 0, so to speak, but the cellular age is already 10 years old, right? Because the telomeres that it inherited was already quite short. And so as the cells in Dolly divides a few more times, right, it would become senescent very soon, much sooner than compared to a naturally born sheep, right? Okay, because those telomeres would have already shortened for 10 years. So maybe it's going to be able to have an additional, 10 or 15 more division as opposed to 40 divisions, right? And that's why it wasn't able to live for a very long time. It died prematurely because all the cells is basically cellularly old. Does that make sense? Okay, so like if people were to, take a cell of me and then put it in an egg and then try to clone me, I'm not sure why anybody would want to do that. But if they were to clone me, then the new mini Vincent that's being born, right? Even though it's a baby, but cellularly it would have the same age as me, right? Okay, and it also will not be able to live as long as one would expect. Certainly not compared to like a normal human, right? It would just start at the age of, say, 40, right? And then the cells would not be able to divide too many times after that. Okay, make sense? So for those of you who are thinking really hard out there, okay, you might wonder, well, how can we reproduce then? How can we have kids if we have kids at let's say age 30? Does that mean the sperm and egg that we pass on to the next generation to create the baby, the cellular age would begin at 30, right? Like, how does that even work? Well, it turns out in the germ cells are the cells that will become the sperm and the egg, right? It turns out in those cells there is a special enzyme that is called telomerase. And the job of telomerase is actually to reset the telomere's length before chromosomes are copied into the sex cells. Okay, so every time you produce a sperm or every time you produce an egg, the telomerase will re-lengthen the telomeres. So even though, let's say you're 30 years old, your chromosomes are shortened, right? The telomeres gets to this particular length, but when you make the sperm or when you make the egg, thanks to the telomerase, it would reset it back to the original length, so that when you pass it on to the next generation, they have a fresh start. They don't run into the same problem as Dolly because of the telomerase. Okay. Now it was a problem for Dolly because we didn't use the sperm and the egg nucleus for the genetic material. We took it directly from a somatic cell, which doesn't have the telomerase, and that's why it started with shortened telomeres. Okay, but again, that's not a problem for normal reproduction because of the telomerase. So without the telomerase, it gets shorter and shorter and shorter, and then you won't be able to divide anymore. Okay, with the telomerase, you maintain the length, and you are able to pass on the genetic material at their full length when we reproduce. So some people might ask or might think, well, if telomerase can do that, why don't we just get telomerase to turn on in all of our cells? Then we can allow the cells to keep on dividing and not get old, right? Wouldn't that be a good thing? It turns out it's not a good thing. Telomerase does get turned on in somatic cells sometimes, but that usually will turn a cell cancerous. Okay, when the cell divides in an uncontrolled manner, that basically is what forms a tumor, and that tumor can be cancerous. You remember we talked about the HeLa cells, right? In lecture 3, maybe. That was the cancerous cells that was taken from the patient with the ovarian cancer, and they were able to grow it in a petri dish forever, right? It's still growing nowadays. People still use those cells for research, right? So how come the cancer cells can divide virtually indefinitely, right? Shouldn't the telomeres get shorter and shorter and then stop the cancer cells from dividing? Well, the answer to that is cancer cells, they have the telomerase. You're not supposed to have telomerase in a regular cell, right? I told you it was supposed to only be found in the germ cells. But in cancer cells, because of mutation, the telomerase is activated. Okay, and now the cancer cells keep on dividing. It doesn't have to worry about the shortening of the telomeres because the telomerase would just reset it every time it divides, reset it every time. So that allows it to divide basically indefinitely. So some of the researchers are trying to exploit this telomerase aspect of cancer. If they could come up with some telomerase inhibition, then they could stop the cancer cells from, resetting the telomeres length, and hopefully the cells will be prohibited to divide. But it's not as straightforward as it is because, let's say they have telomerase inhibition therapy. The person might become, they might not be able to have kids afterwards because, that would also affect the germ cells, right? And so, there is always a balance that we have to study more before they could come up with a new therapy. As of now, there are no therapy out there that involves telomerase inhibition. Okay, so that was a lot of talking. Let's see how much you have learned. It's much easier to do polls here on Zoom. So on your screen, you should see a poll screen. You can try to pick the answers, please. Okay, I only have 3 responses so far. I'm gonna wait a little bit to see if there are more people. Okay, thank you for those who have participated. Let's take a look at this. What is the key difference between DNA replication in prokaryotes and eukaryotes? Prokaryotes have telomeres and eukaryotes do not. That is not true. Okay, it's the other way around. Okay, we are the ones who need the telomeres because we have the linear DNA, right? We're the ones whose DNA gets shorter and shorter every time. So we need the telomeres. Prokaryotes don't need them because they have circular DNA. Prokaryotes chromosomes have a single origin of replication, whereas eukaryotic chromosomes have many. That is the correct answer. Okay. Due to the size and the length of our genetic material, multiple origin of replication is needed to make the process more efficient. Okazaki fragments are found in both eukaryotes and prokaryotes, and primers are needed for both types of cells as well. So the answer is B. Let's try another one. This one should be easier. Go ahead, please. Okay, so to join the Okazaki fragments together, we're going to need the ligase. Okay, ligase is the one that connects them together. Let's do another one. Okay, anyone else before I close the poll? The correct answer is polymerase 1. Right? So remember, as polymerase 3 approaches the primer, it will fall off, and number 1 will continue on and replace the primers with the correct nucleotides. I think there's one more. Yeah. Just very quickly, let's do this one. All right. Thank you. I'm going to end the poll now. So telomerase is the enzyme, right? ASE, that's the enzyme that's responsible for resetting the telomere length, right? So A, re-lengthen, shortens chromosomes in some cells. That's the correct one, okay? Some cells being the germ cells, right? Does telomerase prevent cancer development? No, it does not. In fact, if a cell other than a germ cell expresses telomerase, it's likely to cause cancer. So that's not it. Remove non-coding repeats of TTA-GGG. It does not remove them. It actually adds them, right? By adding more TTA-GGG, you reset, you re-lengthen the telomeres. Do you need the telomerase to shorten the ends of the chromosome? No, the chromosome shorten on its own every time it divides. We need the telomerase to reset them, to re-lengthen them. So that's not correct either. Right? So it's not all of the above. The correct answer is A. One person got that right, whoever you are. Good work. And the rest of you, hopefully I have convinced you why the answer is not E. Okay. Moving on. Next, we're going to talk about the cell cycle and mitosis. Now, in lecture 3, we learned about the cell theory. So here are the 4 parts of the cell theory again. Every living organism consists of 1 or more cells, and the cell being the smallest unit of life. And then the 3 and 4th point here, 3rd and the 4th point here is what we are focusing on in this particular lecture. The 4th point, cells contain hereditary material which they pass to their offspring during division. That is basically what happens with the DNA replication, right? That's the whole reason of why we replicate our DNA, why we copy our DNA, so that we will be able to pass it on to the next cell when they divide. Okay, and the 3rd point here that says all living cell comes from pre-existing cells. Right, so this is the mitosis. The cell division part is called mitosis, and that's what we'll be talking about. Okay, but it's important to remember DNA replication occurs before the mitosis. You cannot divide your cell before you copy all your genetic material. So this must happen first, followed by mitosis. Why do we need cell division? Okay, what's the purpose of dividing the cell? Well, here are 3 main reasons of why we do it. Number 1, replacement of lost or damaged cells. If you have a cut, you are likely to damage some cells. You might have even lose some cells, and those cells needs to be replaced. I mean, how are we going to get more cells? Where do we get the replacement cells from? Well, they would come from the division of neighboring cells. Okay, so all those cells that are near the wound, they can divide and fill in the wound and replace the lost or damaged cells. Second reason of why we do cell division is for growth and development. The number of cells you have right now in your body is a lot more than what you had when you were a baby. All those extra cells, they had to come from somewhere, right? And again, it came from the division of existing cells. Finally, there is reproduction. Right? Some organisms rely solely on cell division to reproduce. Okay, humans, we don't do it. Animals, we don't do it. Okay, animals, we do. Well, most animals don't do it, I should say. Okay, most animals require sexual reproduction. Okay, which means 2 separate individuals are needed. Each individual contribute half the genetic material, and that creates an offspring. But for some organisms, they do mitosis, okay, which is just cell division, and that is what we call asexual reproduction. So alone, just by using mitosis alone, they are able to reproduce. Okay, so in this kind of division, in this kind of reproduction, asexual reproduction, no fertilization is necessary. No egg, no sperms are needed, and you only need 1 individual to do it. So that's an advantage, right? Sometimes it's hard to find a mating partner in nature, right? Because the organisms don't necessarily live close to each other, and it could be very difficult to find a mating partner. And so if the organism requires just itself to reproduce, mitosis, then they can reproduce basically anywhere, right? And it's a much faster process, and it requires less energy. Okay, so to put it in the context, right? A bacterium like an E. coli, it could divide, every 20 minutes on its own, right, without a partner. But for like humans, in order to have a baby, right, it takes like 10 months, right? So the a process is usually much faster for asexual reproduction. Now, it doesn't have to be restricted to like single cellular organisms like the amoeba here and the hydra. Big organisms that have thousands and thousands of cells can do that as well. So sea star can do that. Did you know if you cut a starfish in two, each of the half will become an individual starfish? They'll be clones of each other, okay? But they practically would be divided into two individuals that used to be one. Okay, and again, that has advantage to it, right? A lot of plants, they do that, right? You break off a piece of plant, then you put it in some water, then it starts to grow its own root, and then you can replant it in a separate pot. And then now you have two plants that are genetically identical. So these are examples of asexual reproduction. Okay, the disadvantage is low genetic diversity. Okay, every individual that came from the original one would be genetically identical, and so if there is some kind of unfavorable environment that targets this particular gene genetic makeup, then all the individuals would suffer. Okay, there's no genetic variation within the population to withstand such environmental pressure. Okay, but we talk about this concept in lecture 1, the idea of evolution, right? Okay, if the organism is able to reproduce really fast through asexual reproduction, then every time it copies its DNA, there's a chance of making a mistake. Okay, and if sometimes that mistake creates a favorable genetic outcome, right, then that could drive the evolution within the population. Okay, but generally speaking, right, low genetic diversity is a major disadvantage of asexual reproduction. So just want to go through a few terminologies here, just to make sure that we're all on the same page before we continue with the discussion on mitosis. We learned about DNA all the way back in lecture 2. We know that it's a double helix and it's made up of these A, G's, and T's. Right? But the DNA is really, really long, and it's very difficult, impossible to pack all of our DNA into the nucleus of a cell. Right? So we must take the DNA and we must wrap them around proteins to make them more compact, to make them more condensed before we can fit it into the nucleus. And so when you wrap the DNA around proteins, you get what's called chromatin. Okay, these are more compact version of the DNA. And when you are about to undergo mitosis, when you're about to do the cell division, we need to take the chromatin and condense it even further so that we can move them around in the cell without having to worry about tangling them up and breaking them. So when you take chromatin and you condense it even further, you would end up having chromosome. Okay, so chromosome is essentially the most condensed version of DNA. Okay, and DNA is just on its own a free form that's not compact at all. Right. So in a non-dividing cell, in a cell that's just doing its own thing, performing its regular function, its genetic material will be found in the form of chromatin. We learned about this in lecture 3, actually. But when we are about to divide, when the cell is about to undergo mitosis, then the chromatin would condense and it would become chromosome. Okay, so these two, again, it's basically the same stuff. The only difference is how condensed are they? If it's really, really condensed, it's chromosome. If it's medium condensed, we call it chromatin. So in different organisms, there are different number of chromosomes, and I've asked you guys in class a couple times before how many chromosomes we have, right? Hopefully you know by now that humans have 23 pairs of chromosomes, a total of 46, and half of it would be inherited from mom, and the other half would be inherited from dad, right? So if we talk about the 23 pairs of chromosomes, you would have like chromosome number 1. Okay, the longest one is called chromosome 1. Okay, so chromosome 1 from mom, chromosome 1 from dad. And then the next longest one would be chromosome 2 from mom, chromosome 2 from dad. And the next one, chromosome 3 from mom, chromosome 3 from dad, so on and so forth, all the way up to chromosome number 22. Okay, so all these chromosomes, number 1 through 22, oh my goodness, 1 through 22, these are called autosomes, okay, or autosomal chromosomes. These chromosomes determines everything about you except for your sex. Okay, so it determines things like your eye color, your blood type, whether your earlobe is attached or detached. It determines whether you have curly hair or straight hairline or widow peak, right? All these features about you, they are all encoded within these autosomes, the first 22 pairs of the chromosomes. Okay, and then your sex would be determined by the sex chromosome, which is number 23. Okay, so for male, chromosome number 23 would be X and then Y. Okay? Whereas for female, both chromosome 23 would be X and X. So XX for female and XY for male. Excuse me. Who's responsible for determining gender of the child? Is it the father or is it the mother? Who's responsible for determining the gender, the sex of the baby? That's right. It's the dad, right? Because the dad has the Y chromosome. If the dad passes on the Y, then it's going to be a boy. If the dad passes on the X, it's going to be girl. I mean, the dad doesn't. He cannot willfully choose it. Okay, it's not up to him. It's all by chance. Okay, but genetically, it's the male that would determine the sex of the baby, depending on which chromosome they passed on. My grandmother, it's like in her 90s now. She's very traditional. So every now and then she would ask me, it's like, why does your wife give you no son? You have 2 daughters. You have to keep trying, have another son. Tell your wife she has to give you a son. And then I would try to tell her sometimes, like, it's not her fault, Grandma. It's actually my fault. I am the one who is supposedly to determine the gender of the child. But she's not having any of it. I just want to make sure that we are all on the same page, that it is the male, the father, that is genetically responsible for the gender of the child. Okay. Size does not correlate with the number of chromosomes. We have organisms that are much smaller than us, right? Hedgehog and goldfish. They have way more chromosomes than us. Okay, there's no correlation between size and the number. It's just sometimes they might have like filler genetic material that doesn't really do anything, right? All right. That takes us to 1:30. Why don't we take a 5-minute break and we will resume afterwards. See you all in a bit. All right, let's keep going here. So the next slide here, it talks about the cell cycle. Sorry, 1 second, please. Okay, the next slide here talks about the cell cycle, which is the interface and mitosis. So cell cycle basically is talking about the life of a cell. Okay, so for most of its time, a cell is just doing its own thing, okay, performing its normal function. If the cell is supposed to be making hormones, then that's what it's going to be doing. If the cell is supposed to be, producing energy, that's what it's supposed to be doing. So all these normal functions are carried out during the interphase, okay? Now, along the way, the cell will also be growing, getting bigger in some cases, and they will be preparing for the division, preparing for mitosis. Okay, so this would be the longest phase of the cell cycle. Now, some cells, they don't actually divide at all. They are stuck in the interphase, and they would never divide. Does anybody have an example of a cell that they know about in the body that actually does not divide? Anyone? Think about it. Which cell in the body that if you lose it, that's it. You're not getting new ones. No? Never heard of anything like that before? Okay, yeah. So for Tomato say neurons, that's right. So a lot of brain cells, they don't divide. You lose them, they don't come back, right? And another example of that would be like heart cells. Okay, people who suffer from heart attack, they don't get new heart cells. Okay, heart cells, neurons, right? Don't divide. So they will be in interphase their entire life. Okay, but for many other cells, they would divide. Okay, and some cells would divide more often than others, like your skin cell, for example. You replace them in matters of weeks. Okay, but other cells they would take much longer to replace. Some organs are remarkable in its regenerative capabilities. Does anybody know of an organ in the body where if you donate, let's say 1/3 of it to someone, you would grow it back? Anybody know what organ that might be? What organ can you donate 1/3 of it and you will just grow it back? No, it's a kidney. Yeah, good try, good try. Well, we have 2 kidneys. We can donate one, but unfortunately, we cannot grow the missing one back. But yes, the liver is remarkable in its regenerative capability. Okay, you can lose quite a big chunk of the liver, and we just grow back in months. Okay, so very rapid mitosis there. So, yep, the mitosis is the actual cell division, and the parent cell becomes 2 daughter cells. Over here, this shows the chart, right? So within the interphase, there are 3 separate stages. Okay, so in G1, it's kind of like getting your driver's license, right? G1 and G2. So G1, that's where the cell will be growing, gathering resources. And then there is going to be a checkpoint here. We're going to talk more about these checkpoints later. But basically, you're going to have a checkpoint right before you copy your DNA. And then you're going to have another checkpoint right after you copy the DNA. Okay, so let me just briefly explain what they are. Over here, before you copy the DNA, there are several things you want to check. For example, you want to check to see if the cell is in healthy condition. Okay, you want to check to see if there are any DNA damages that were not repaired. If it's not repaired, maybe you try to repair them. And if it's not successful, then you will not allow this cell to go forward and proceed to division, right? You might want to check the telomeres length. If the lengths are critically short, then it will not be allowed to proceed either. And sometimes if the cell is just too old, you can't keep them around anymore, then they might trigger apoptosis, right? Which is the programmed cell death. So that's what this checkpoint is for, to check for the quality and the condition of the DNA in the cell and make sure it is healthy enough to proceed to the actual DNA replication. Once you copy all of its genetic material, you want to do a second check, okay, which is to see if the DNA replication process was completed. Okay, did you copy everything? Did you make any mistakes when you were copying it, right? What's the quality of the DNA replication? Right? Is everything okay, basically? Did you do a good job copying it? And if everything's okay, then it's able to proceed to finish with the final preparation for division. Then you go into mitosis and you will end up having 2 cells. Okay, so if along the way a cell fails checkpoint 1 or checkpoint 2, it will not be permitted to go on to mitosis. More on that later. So here we have a cell that is in interphase. That's the growing phase, right, where the cell spend most of its life in. This is not part of the division yet. This is a real picture of a cell that is in interphase. The blue shows you the DNA and where the chromosomes are. Right now, you can't really see the chromosomes because they exist in the form of chromatin, right? Okay. The chromosome only shows up when you start doing the division. Okay. Right now, they're just in a condensed form, but not the most condensed form. We have some yellow to show you the fibers that will guide the movement of the chromosomes, and the red just kind of shows you where the cytoplasm is. So in the interphase, right, the cell is performing its regular function, and towards the end of this phase, the cell will prepare to undergo mitosis. Okay, and also during the interphase, which again breaks down to G1, S, and G2 phase, right? In the S phase of the interphase, that's where the DNA will replicate itself. Okay, so all the stuff we talked about, what the gyrase and the helicase, all that stuff happens in the S phase of the interphase. All right, I gotta go through a few terminologies here before we proceed, and it might be a little bit confusing, so please do ask questions if you are not following. Here we have one chromosome and then we have another chromosome. Okay, this one is, let's just call it chromosome number 4 from mom and chromosome number 4 from dad. Okay, so this is the maternal one, and then this is the paternal one. Okay, and you would have again, right, like 22 pairs of these, right? Chromosome 1, 1, 2, 2, 3, 3, 4, 4, all the way to 22 and 22, and then XY for male and XX for female, right? So these two chromosomes, they are the same length and the same size. We call them homologous. Homologous chromosomes. Same length, same size. Okay, but not identical. Okay. Our parents are not genetically identical, right? Thank goodness, because they have different features. Okay, so like on the mother chromosomes, right? There might be a gene that codes for like brown eyes, and on the other chromosome, right, it might also be brown eyes because both of your parents have brown eyes, and that's totally normal. But on another location, your mother might have like curly hair, which I'll represent with small c, but your father might have straight hair, right? So these are gene that codes for the same information, but not necessarily the same version of that characteristics. Okay, so they both code for hair type, but one is for curly hair, the other is for straight hair, right? You might have another gene here, right? One might code for like type A blood, the other might code for like type B blood, and that's going to be different. And then maybe another one, right? One codes for like, I don't know, totally making this up, attached earlobes, and then the other one also happens to be attached earlobes, right? So along the entire length of the chromosomes, you have the genes in the same order, but the version of the gene might not be the same. Okay, so these chromosomes, we call them homologous chromosomes because they are the pairs, right? Chromosome 4 and chromosome 4, they are homologous. 5 is homologous to 5. 6 is homologous to 6, okay? 7 is not homologous to 8. It has to be the same number for them to be homologous. So I ask you, and please type your answer in the chat box. How many homologous chromosomes does men have? Okay, let's do men first. How many homologous chromosomes are in men? And this is true for all men, okay? How many homologous chromosomes are there for men? Anybody? Okay, we have one answer, okay, from Bree22. Anyone else? Anyone have another answer, or everybody agrees it's 22? You guys are quiet in class, but now you're extra quiet online. 22 is correct, okay? Because men is XY, right? X is not homologous to the Y, okay? So 22 homologous pair for male, 23 homologous chromosomes for female, because XX, they are homologous, right? Are we together? Are the rest of you like with me? Okay, thank you for the thumbs up. I am going to ask another question. Okay, this is a straightforward question. You have to try to answer, because I want you to get this wrong so that you remember not to get it wrong on the quiz. Okay, so please participate. First question. No one will get this one wrong. Okay, please participate. Okay, this one. How many chromosomes do we see here? Before we do the replication, how many chromosomes are here? Okay, it's not a trick question. Okay, yeah. Good. Yes, I see a few 2s popping up. Okay, good. Yeah, there are 2 chromosomes here. Yes, 2 chromosomes. Okay, again, chromosome is just like really condensed, really easy to move them around, very easy to see them. And then you're gonna do DNA replication. Okay, so you're gonna make an exact copy of this maternal number 4. So now you're gonna have number 4 from mom and number 4 from mom, and they are exactly the same as each other. Okay, and they will be connected. They will be glued together at this location called the centromere. Okay, centromere. So you chromosome 1 from mom, chromosome 1 from dad, and then after DNA replication, you will have 2 chromosome 4 from mom and 2 chromosome 4 from dad, and then the ones that are exactly the same will be stuck together at the centromere. Okay, so these copies that are exactly the same as each other, we call them sister chromatids. Sister chromatids. Okay, sister sticks together. That's what I tell my girls, and that's how you can remember them. The sister chromatids stick together at the centromere. Okay, so that's chromosome 4 from mom, chromosome 4 from mom, chromosome from dad, chromosome 4 from dad. Okay, these are identical copies. Identical copies. Okay, these are not identical copies. They are homologous chromosomes. Okay, so let me ask you again. We started with chromosomes. After DNA replication, how many chromosomes do we have here? How many chromosomes do we have here? Please type the answer in the chat box. How many chromosomes? Ow, I just put on my headphone. Okay, I have one answer so far. Okay, a few more. Okay, come on, just type your answer. Type it in. Get it wrong right now so you won't get it wrong on the test. Is that all? Is that everybody? Are the rest of you like here? Or you just turn on the Zoom and went away, huh? Okay, so we have a bunch of answers. We have some people say 4, some people say 2, some people say 16. All right. So this is very confusing. It was confusing for me for the longest time. The correct answer is 2. And you're like, what? Right? How is it 2? Well, biologists, the people who came up with this stuff, they've decided that as long as they are stuck together, we will still count it as 1. Okay, so there's one chromosome here and two chromosomes. Okay, but there are four chromatids in this case, right? You see the difference? So as long as they're stuck together, we'll still call it one chromosome. But this chromosome has two chromatids. Okay, this chromosome is just chromosome. There's no chromatids because you haven't copied anything yet. Do you understand that? Please remember it, okay? Especially when we talk about the mitosis thing, knowing how to count the numbers is going to be important. All right, moving on. This is the actual mitosis. We have four phases, and you can memorize it with this acronym, PMAT. PMAT, prophase, metaphase, anaphase, and telophase. So I'm going to go through what happens in each of these phases, and hopefully it will make sense. Starting with early prophase. Okay. Now, you have to remember, let me just write it out for you again. Cell cycle, right? Cell cycle, it's separated into the interphase. And interphase is separated into G1S and G2, right? G1, S, and then G2. And if you pass all these, if you pass G1, you finish your S and pass your G2, you will be allowed to do mitosis. Okay, so mitosis is separated into the PMAT, and we are talking about the prophase now, which means if the cell is in early prophase, it would have already gone through G1, S, and G2. Okay, that means the DNA would have been copied already. Okay, so in the early prophase here, chromatin coils into visible chromosomes. And you can see the individual chromosomes now, right? They're kind of like little noodles, right? Okay, you weren't able to see that before. Let me just quickly go back to show you an earlier picture over here. When the cell was still in interphase, you cannot make out the individual chromosomes because they are still in the form of chromatin. They have not condensed further yet. Over here, it is very condensed. It has become the chromosomes. Okay, the whole point of making them into chromosome is so that you can move them around easily. So by this point, DNA replication has completely finished. The sister chromatids are held together at the centromere. Okay, so there you see a centromere here. That's one sister chromatid. That's another sister chromatid, right? Okay, you might have another centromere here, another centromere here. Okay, and all these replicated sister chromatids are stuck together. We need to get these chromosomes out of the nucleus, so it's a good time to dissolve the nuclear envelope, okay, to make it disappear. And the centrioles, which you might remember learning in lecture 3, they will produce the spindle fibers. Okay, that's these cables that will guide the movements of the chromosome later on. So all this is happening at the early part of the prophase. By the time we get to the later part of the prophase, the nuclear envelope will be completely dissolved, right? So you see the chromosomes are all outside now. Over here, they are still restricted within a circular boundary, although the nuclear envelope is starting to disappear. You can see these two yellow dots. It looks like the eyes here, right? Those are the centrioles. Okay, but by the time we get to late prophase, the chromosome is out of the nucleus. Okay, and we have the centrioles at the opposite side of the cell. Okay. The spindle fibers, the cables that we're pulling the chromosomes, right? The spindle fibers will be attached to the centromere of each chromosome pair. Okay, so here we have sister chromatids stuck together at the centromere, and you will have one spindle fiber from one side and other spindle fiber from the other side. Okay, and all of them would be like that. And that's it for the prophase. Okay, so P is for preparing. Okay, all this thing that has happened is to prepare for, the separation of the cell. After the prophase, next comes the metaphase. M is for middle. M is for middle. Okay, in metaphase, the spindle fibers from both sides will be pulling back and forth in a tug of war that will align the chromosomes all in the middle of the cell, like this. Okay, so all the chromosomes will line up. Now, there are only 4 chromosomes here to show for simplicity, right? In humans, we would have a total of 46 of these lined up in the middle. It would be too busy to draw all 46. So there's just 4 here. Okay. After you line them all up in the middle, the next thing to do is anaphase. A for apart. You're going to be pulling the sister chromatids apart. Okay. So how many chromosomes do we have in this picture? Well, now that they are no longer stuck together, we would count them separately. So 1, 2, 3, 4, 5, 6, 7, 8, right? So if you have 4 chromosomes in metaphase, you will now have 8 in anaphase. Do you understand? When they're stuck together, you count them as 1. Once you pull them apart, then you count them separately. Okay? This one here and this one, they are exactly the same, right? In fact, these 4 chromosomes are exactly the same as these 4, which makes sense because half of it would go to one cell, the other half goes to the other cell, and you want them to be exactly the same. After all, that's the purpose of the mitosis, right? So that's anaphase. After anaphase, we will have the last phase, right? Telophase, T for tearing. Okay. In telophase, we will basically undo everything that we did in the prophase. So now, after we're done pulling the chromosomes back and forth, they are going to uncoil back into chromatids. We will put them back into the nucleus. So the nuclear envelope starts to form again. Starts to form, not from, sorry. The spindle fiber starts to disappear. We no longer need to pull the chromosomes apart. And then the cell will split in the center in a process called cytokinesis. And the best way to visualize what cytokinesis is like is think of like a big ball of Play-Doh. Okay, you have a big ball of Play-Doh. You're going to tie a rope around the center and you pull on the rope. Okay, that will cut the Play-Doh into 2 balls, right? Yeah, that's exactly what's going to be happening in the cell. Okay, you're going to have this ring that forms in the middle. Okay, the ring will cleave into the cell, causing it to become 2 separate cells. This process, again, is called cytokinesis. All right. So that's it, the 4 phases of mitosis. Now let's quickly go through these ones and see if you can identify it. Okay. What would you say this cell is? This is onion, by the way. Okay, the root tip of an onion skin. What phase would this cell be in? Is it, prophase, metaphase? What do you think? Let's just do a quick poll here and see if you guys are with me. Okay. Okay, is it prophase, metaphase, anaphase, or telophase? This first one right here. What phase is it? This one. Okay, I didn't get too many response, and for those of you who participated, thank you. And the answers are kind of all over the place. So this one over here, I mean, you can see all of them are right in the middle, right? This whole thing is a cell, right? And then all the chromosomes are in the middle. So if it's in the middle, then it's metaphase, right? Metaphase. This one, you can see the chromosomes are being pulled apart. Okay, you can see the individual sister chromatids moving to opposite poles. So that would be the anaphase. Anaphase. Over here, right, the chromosomes are very concentrated at the end. They are almost going back into the nucleus. And if you look closely, there is a wall that's forming in the middle that's about to divide a cell, right? So this would be the telophase. This one here, you just have a single nucleus. The chromosomes are starting to show, but they're still inside the nucleus. The nuclear envelope hasn't dissolved yet. So that would be prophase. Okay. All right, just try a few more. What about this one? What do you think this one is over here? Just type it out or yell it out. What do you think this one is? Anybody? Yeah, thank you, Azizi and Nelly. This is the anaphase, right? And this is another anaphase. Okay. For these one, they're just in their interphase, right? Interphase. They're not doing anything. I mean, they're not dividing. That's what I mean, right? Okay. That's that. Okay, we will quickly go to the study guide here. All right, let's take a look at this. These pictures, we will label them. Okay. Why don't you try to label that on your own, and we'll take it up on Wednesday. Speaking of Wednesday, I actually just got the I just saw the tweet from the school board. Looks like they will be resuming school on Wednesday. They will be resuming tomorrow actually. So for Wednesday, we will return to in-person learning. Okay, we'll post an announcement as well. So I'll be at the college. We all go back to school, okay, on Wednesday. And that means you have to hand in your lab on Wednesday as well, right? The beetroot lab. Okay. But yeah, I'll post an announcement after class as well in case you forget. I'll also send you an email, right? But again, just to be clear, for the record, we will be going back to the college on Wednesday. Okay. So we'll take this up on Wednesday when we get there. Let's just wrap it up with a few more slides. Okay. So I have been telling you that in the interface, right, there are various checkpoints, right? So there's the G1, and then there's a checkpoint here, checkpoint. And then if the DNA quality is good, you will be allowed to copy them. If it's not good, you will try to repair it. And if it's not repairable, then the cell would not be permitted to go forward. You might even be able to trigger apoptosis, like cause it to die. And after the DNA replication, you will have a second checkpoint. Second checkpoint. Okay. And to see if the quality of the DNA replication was good. If you made any mistake, you try to correct it. And if all is well, then you are able to proceed to mitosis. Okay, so now we are going to focus on the checkpoints and what happens if there is something wrong with these checkpoints. Okay, so let's take a look at the next slide first. Let's do the polling here, and then we will continue from there. Okay, go ahead. Okay, anyone else before I end the poll? Okay. Which phase of the mitosis does the nuclear envelope dissolves? It will actually dissolve in the prophase. Okay, because you need the chromosomes to come out, right? Okay, it would already have dissolved in all these steps. Okay, so it's the first, it's the prophase where the nuclear envelopes will first dissolve. Let's try the next one here. And let's see if you're able to get this right. Okay, so if you have 8 chromosomes at metaphase, right, that means you have 8 of these things lined up in the center. 1, 2, 3, 4, 5, 6, 7, 8 lined up in the center of metaphase. And anaphase, they will be separated, right? So this one will get pulled apart, right? And then this one will get pulled apart. And actually, in the end, right, afterwards, you will have 16. Okay, you only count them as one if they are stuck together. Once you separate them, you have to count them separately, right? So the correct answer is 16. Make sure you understand this. You can think about this one. We don't have time to go through it. We'll talk about it on Wednesday. I want to spend the last couple of 10 minutes or so going through 3 more slides. Okay. So the checkpoints before the S and after the S phase are regulated by proteins. Okay, and of course, proteins, they are encoded by genes. So if there is something wrong with a gene, there could be something wrong with the protein. If there's a mutation in the gene, then the protein might not function properly. Okay, and typically, for the checkpoint proteins, there are 2 types. Okay, we have those that are called growth promoters. Okay, and the function of growth promoters is to promote mitosis. It allows the cell cycles to go forward. They are the ones that gives the green light, the go ahead for the cell cycle to continue to spin. It gives the permission for the cell to divide. Okay, so we'll call these like green light protein. Okay. So think of it, visualize it in your head. If you see a green light, you are allowed to go. On the other hand, we have growth inhibitors, which inhibits mitosis. These are what we call red light proteins. Red light proteins. Okay. And they are responsible for turning on the red light and telling you to not go. You're not allowed to divide. And a cell is not allowed to divide for various reasons. I already mentioned this a couple of times. Maybe the DNA is bad quality. Maybe telomeres are too short. Okay, maybe the cell is in poor health condition. All of this will trigger the red light, and it will not be allowed to proceed with the cell cycle. Okay, so there are green light proteins that will allow the division to occur. There are red light proteins that will stop the cell from dividing. Okay, the proper names are growth factors and growth inhibitors. But please keep in mind the image of red light and green lights. It will help you understand the following slide. Okay, so proto-oncogenes. Proto-oncogenes are genes that codes for green light proteins. Okay, all these proteins has a gene, right? So the gene that makes the green light proteins, they are called proto-oncogene. So the proto-oncogene normally would help the cell grow by turning on the green light. You're allowed to divide, you're allowed to make more copies of yourself. However, when a proto-oncogene mutates, when there is a mistake in the gene, right? Or sometimes if you have too many copies of it, and that could happen, I'll explain how later on in the course. If you have too many copies, or if you have a mutated copy of the proto-oncogene, then it becomes a bad gene, okay? And in this bad form of the gene, the protein that it codes for, the green light proteins, will be permanently turned on. Okay, it will permanently be activated. So think of the green light and the red light, right? In this case, because of the mutation, the green light is always on. If the green light is always on, then the cell will always be going through the cell cycle. It will keep on dividing and dividing and dividing, even when it's not supposed to. Okay? Because the green signal is always on. Okay? So this out of control cell division is often what leads to cancer development. Okay? So the bad gene that we have now is called an oncogene. Okay? So proto-oncogene is the original normal version. It's the healthy version. Nothing's wrong with it. It promotes cell growth. Okay, but it doesn't turn on the green light all the time. It just turns the green light on when it's supposed to. Okay, but when it mutates to an oncogene, then the green light is always on. The cell is allowed to divide all the time, regardless of whether it's supposed to or not. Does everybody understand the concept of the proto-oncogene and the oncogene? Remember, the oncogene is the bad one. The proto-oncogene is the normal one. Okay. Next, we have the where's the other one? Did I miss a gene? Oh, how come the slides are okay? We'll skip this slide for a second. We'll just go on to this one for a second. On the other hand, we have what's called tumor suppressor gene. Okay, so tumor suppressor gene, they code for the red light proteins. Okay, so these are genes that codes for proteins that normally would slow down cell division. Okay, they are the ones that repair the DNA when there's something wrong. They will tell the cell to die when it's too old, when it's not supposed to divide. They are the red light that stops the progression of the cell cycle. Okay. However, if you have a mutation that causes the tumor suppressor gene to not work, then in this case, the red light does not turn on. And you have to understand the difference of what I'm saying. For the oncogene, green light is always on. Okay. For the tumor suppressor gene, when you have a mutation, red light does not turn on. Okay, so either way, if the green light is on all the time and the red lights are not turning on, then the cell will keep on dividing and dividing, right? And that's what gives you the cancer in the end. Okay. So for the proto-oncogene, remember we have two sets of chromosomes, right? One from mom, one from dad, and on each of them you would have a gene. So I would have a proto-oncogene on each of the chromosomes. However, only one of them needs to be mutated to cause trouble. Only one of them needs to have the mutation to cause the cell to divide in an uncontrolled fashion. Okay, so think of these two genes as the two green lights, okay? So that they will turn on and sometimes they will turn off depending on whether the cell is allowed to proceed or not. However, if one of the copy is messed up, then you would have a green light that's always on. Right? The other one might be functioning normally, turning on, turning off, turning on, turning off, but it doesn't matter because you have one copy that's always green light, and that's what the cells will follow. And then it will keep on dividing and dividing, despite the fact that you have a normal functioning version of the gene. Okay, so only one hit one mutation on one of the copies is enough to push the cell over the edge and cause it to divide in an uncontrolled fashion. Think of it as a car with a stuck accelerator. It would just keep on going and going and going and doesn't know when to stop. Okay, we call this a gain of function mutation. Okay, because before it was being able to turn on and off, but now it's stuck on the on function, right? So it's gain of function. It's overworking. On the other hand, the tumor suppressor gene is a loss of function. Okay, so think about, again, 2 chromosomes and one tumor suppressor gene and another tumor suppressor gene. Let's say you mutated this one. So this red light does not work. But that's okay. You still have the other red light, right? As long as the other red light still knows how to turn on and off, you can still stop the cell from progressing. Okay, it's only when both copies of the tumor suppressor genes are mutated, then you have no more red lights, and that's why the cell cycle will progress. Okay, so it is easier for a cell to become cancerous if the mutation happens in a proto-oncogene. Okay? However, if it's a mutation in a tumor suppressor gene, it would need to happen 2 times in the same cell in order for it to cause a problem, right? Okay, in the same cell though, right? Like you have trillions of cells. If you have a mutation in a tumor suppressor cell gene in one cell and then have the same mutation in another cell, that's okay because each of these cells will still have one functioning copy, right? It's only if you have both copies that are mutated within the same cell, then you start to grow in an uncontrolled manner. Okay, we'll come back to this point later on Wednesday. People born with only one functional copy are more likely to get cancer. I'll explain what that means later. Okay, that's where we're gonna leave the PowerPoints. But we still have like 4 more minutes, so let's quickly take this one up right now, so that you can look through it yourself. All right, so over here, it's a plant cell. It's rectangular with the cell wall and stuff like that. We see the nucleus still there. The chromosomes are not quite formed yet. They're still in the form of chromatin. So this would be interphase. Interphase. Okay, let's just do all the plant cells first. This here, now we can see the individual chromosomes. The nuclear envelope starts to disappear a little bit. We see some spindle fibers. That would be the prophase. The sister chromatids are pulled apart here. And so this is anaphase. Over here, everything is lining up in the middle. Middle metaphase. Over here, the chromosomes are on the opposite side. They are starting to form the nuclear envelope again. And then we're trying to cut the cell in half. So that would be telophase. Over here, anaphase again. Let's do the animal cells. We're trying to pull the cells apart. That is the telophase. Anaphase here. Metaphase. And prophase. Okay, I will only give you cartoon diagrams like these ones to label. Not the last one. The last one is not cartoon. The last one is real. Okay, but I would just give you these cartoon diagrams to label on the quiz. The quiz is going to be next Wednesday. I'll see if we have to move it. Depends on how much we covered this Wednesday. I'll give you a final answer on Wednesday. Okay, but as for now, it is going to be the following Wednesday. Okay, so thanks very much for coming. I will post the recording here. And I will see you at the college on Wednesday. If you see your friends, please let them know. Yeah, and I'll post an announcement as well. If you have any question about any of these things, please feel free to contact me. Okay, thanks again. I'll see you all later. Bye.
Thank you.
No problem. Thank you.